Hi, I'm Linh Tran. I have recently completed my PhD in genetics at the University of Arizona. Thanks to the organizers for the opportunity to present Confusion, one of my dissertation projects in collaboration with David Castellano, a postdoc in our lab, and my PI, Ryan Kuhn-Kunz. So the research in our lab lies in the field of population genetics, a field with a long history dating back to almost 100 years ago now. And in recent years, there has been a lot of excitement in the field around the development of new methods based on deep learning algorithms, specifically convolutional neural networks, with reportedly very promising performance for a variety of population genetic inference tasks. But this has also left several unanswered questions about the interpretability of these published models, which we hope to address with Confusion. And while there are a variety of approaches from the field of explainable AI that you'll probably hear about later today, the unique aspect of Confusion is that it leverages the specific domain knowledge of population genetics for interpretability. Its strength lies in taking these powerful but black box deep learning methods and grounding them back to the theoretical foundation that has been established in the field for a century. So specifically, Confusion take the original test data and make a disrupted version of it at different levels of disruptions, where each level can be attributed to a known informative feature of genetic data based on population genetic theory. We can then test the trained networks on these disrupted data set to identify the features that are important to the trained CNN's reported performance. The simple but key insight that we can disrupt the data in an informative way comes from the fact that for many population genetic inference tasks, our input data is a haplotype matrix. As seen in this example here, say we have a sample in a line DNA taken from 3 diploid individuals. We can focus only on the single nucleotide polymorphic sites, which are colored in purple and yellow here. We end up with 4 columns of segregating sites and 6 rows of sample haplotypes. And as a minimal example, let's fill in to have a few more sites in our haplotype matrix. This is the original data from which we can start creating the disrupted versions. So first, we can disrupt the spatial pattern by randomly shuffling the columns to change position of the original sites. Here, for example, we switch column 1 and 8, 2 and 4, 7 and 5, and so on. Next, we can disrupt the linkage disequilibrium statistic, which is this pairwise correlation coefficient R square between pairs of segregating sites. This decay curve of this R square over the genetic distance is informative for many population genetic tasks. So to disrupt LD, we can check individual columns and then randomly shuffle the content within the columns. For example, here, I'm having a schematic that's showing the shuffle of the first column, and then the second column, and then so on and so forth for all columns individually. This operation essentially destroys the original pattern of correlations between the segregating sites, thereby disrupting LD. Another classic population genetic statistic that we can disrupt is the allele frequency spectrum, which is the distribution of alleles at different frequencies in the sample. So in this toy example that I have here, we have 3 frequency classes. The singletons that only appear once, as seen here in columns or site 4, 5, 6, 7, so total 4 sites. Then the doubleton, which appear twice in the sample, here total 3 sites, and finally only 1 site at exactly 0.5 frequency. And to disrupt this AFS signal from the original data, we can simply freely shuffle all the entries in the matrix. So as shown here, this disruption will cause the shape of the allele frequency spectrum to change. Finally, we can go very extreme and generate this sort of non-biologically meaningful data matrix by pushing all pixels of the same color to one side. Now I will show the results of applying these shuffle test data to 3 previously published CNNs in the field. The first one is a discriminator of a generated adversarial networks, or GAN. This discriminator is a CNN that takes in this haplotype matrix shown on the left and classify whether it is neutral or contains signature of positive selection. And the performance here is measured by a ROC curve and the area under the curve value. As shown here is the reproduced results from the original study. So first, disrupting the spatial pattern by shuffling columns, and we can see that the performance does not change very much. Also no change when we disrupt the LD statistic. Same trend that we observed for a disruption of the allele frequency spectrum. And then finally, the performance only seemed to decrease a little with our most extreme disruption. So in this case, we suspect that perhaps the test was too easy to really discern whether the network has really learned any important features from the data. Next, we tested a CNN trained for a multi-class classification task. So this one's quite similar to the binary classification that we shown before, but now we have one additional class. So total of 3 classes: neutral, medium selection with the selection coefficient S value at 200 and strong selection at 400. So the performance here is measured by a confusion matrix as per the original study. Again, we disrupt the spatial genomic pattern by shuffling the columns, and there are virtually no change in the performance. However, when we disrupt the LD statistic, we see that the performance breaks down and gives all neutral prediction. So for this CNN, we can conclude that LD is an important feature that drives the performance. And as expected, performance remains broken as we apply more extreme level of disruption. Finally, we tested a CNN trained for a regression problem, which involves inferring the parameter values for this demographic model that has 2 population side change events shown here. As per the original study, accuracy performance for each parameter is shown as a scatter plot with the true value on the x-axis and the predict value on the y-axis. So we apply our test as before, and as shown, the CNN is not sensitive to the disruption of spatial genomic patterns, and also not sensitive to LD. However, it is sensitive to the change in the allele frequency spectrum, as you can start to see the performance breaks down for the CNN here. So in summary, we have developed Confusion, a method for interpreting the performance of CNN trained for different population genetic tasks. With Confusion, we were able to identify the features that are important for the performance of previously published CNNs in the field. And through this work, we want to highlight the fruitful approach of utilizing specific domain knowledge to improve and inform the adoption of black box algorithms such as CNNs into other domains. With that, I'd like to acknowledge the funding that has made this work possible. Here are my contact information and the GitHub repository for this project. And also, soon will be joining University of Washington Genome Science Department as a postdoc. So I really look forward to connecting with local folks. Thanks for your attention, and I'm happy to take questions.
Any questions?
So I have a question. Great talks. So when you compare these kind of models, have you looked at different kinds of selection? Like, for example, polygenic selection versus like a positive selection, you know, to see whether those different signals you identify here will be different among some of these different selection signals?
Yeah, that's a good question. So in this case, we sort of focus on the published CNN, and the one that I just happened to pick here just have the example of positive selection. So we haven't done it on other models that have been trained for other selection, but it is definitely possible to apply the same method to whichever trained model on a different task that you're interested in to see how does that apply to that different model.
Yeah, thank you.
Thank you.
Hi, thank you for the talk. I also have a question. I didn't quite get, is it possible to see maybe a combination? So if for some variants you disrupt the LD and for some other the spatial location, how will that work out?
What do you mean by combining? Are you talking about combining these different?
Yes, because I guess for some variants or some locations, maybe LD is more important, whereas for some other locations, maybe some other feature is more important.
Yeah, so in our work, that's exactly what we're trying to get at here with this different disruption. For each disruption, we're trying to attribute that to a particular feature, either it's LD or AFS. Within, if I'm getting this right, you're referring to maybe like different location with different importance. And I think for our method, it's quite not really focusing on that. But sort of more focusing in general in just the overall like LD, but not quite like specific to each site or location.
Okay. Thank you.
Thank you.
Thank you for great talk. I have a question about the data leakage. So your method showed some CNN models may have been overfitted. So I wonder if in the tasks that such CNN models addresses, is there any possibility of data leakage in training and test data?
Yes. So I guess technically it is a question for the original paper, because we sort of just take whatever they did and reproduce it and then apply our disruption. I do want to emphasize that for most of these trained models, they were trained on simulated data. So it is not as much of a concern because you sort of can control simulation for the train versus test. So this is unlikely, but there might be other problems.
Thank you.
Thank you.
Yeah, following up on that, I know Sarah Matheson's stuff is trying to address some of that misspecification with the simulations. But so you're training on simulated data. You can simulate different type of evolutionary processes. And have you thought about exploring, training your own models on different type of simulations to see how sensitive your tests are at revealing, those differences?
Yeah, we haven't really thought about that. I think for all of these models, the model that was used to simulate is pretty limited. It's usually like you pick one demographic history and then you just simulate a bunch of data based on that. And then the limitation sort of our test is also the result I'm showing here is very specific to the one test set that was used. And if you simulate a different test set, you might see a completely different pattern. So it's definitely, you need to kind of reapply it to different situations to see a more general trend.